Welcome to Biochemistry. Biochemistry. Before discussing biochemistry, let me introduce myself. And I am Muhammad Imran Sajid, and this is going to be a great session. I will be explaining all the biochemistry that is necessary for FPGEE purpose, and this is basic subject that is necessary not only to discuss the metabolism but that is not normally happening in the body, but also the metabolism of drugs. This forms the basis, right? So let's start. Biochemistry, as you all know, that biochemistry is a branch of science, right? And it deals with two things. Number one, biological molecules, and number two, biological processes. And biology, what are those biological molecules? Let's see. You all are very much familiar. These are the carbohydrates, proteins, lipids, nucleic acids, vitamins, hormones, enzymes, and minerals. Out of these biomolecules, the most important are the four. These are the major biomolecules. These are the carbohydrates, proteins, lipids, and nucleic acids. And nucleic acids are very much important because they are dealt with in a separate branch of biochemistry, and that is called genetics, right? So we reserve it here. So let's talk about these three major biomolecules that are important for energy purposes. These are the three things that give us energy: carbohydrates, proteins, lipids. And now we come to the biological processes. All the biological processes that are happening in the living organisms or in the human being are collectively termed as metabolism. The metabolism can further be classified into two categories: catabolism and anabolism. Catabolism is the breaking down of the larger molecules into smaller molecules, while anabolism is the combining of smaller molecules to make larger molecules. So let's discuss what is this. This is the overview of energy metabolism. And this one diagram, this one page, and this one presentation, this one page is really very much important in understanding the whole energy metabolism. So I want you to remain with me, and I hope you will enjoy it. You see that all the food that we eat, I mean all the pastas, and pizzas, and burgers, and fruits, and vegetables, and meat, all that thing contains these three major nutrients. These three major nutrients are carbohydrates. These three major nutrients are this page. This page is really very important to understand, and this makes the basis of energy metabolism, and I call it the big picture. Unless you know this big picture, you are not going to grasp the whole concept of biochemistry. So this is really very important to spend some time over this page, and this is overview of energy metabolism. I mean, what is all about energy? How do we get energy from the food that we eat, right? Overview of energy metabolism. So we eat in our diet all those pizzas, burgers, pastas, meat, fruits, vegetable, milk, all that thing. All that thing is essentially made up of carbohydrates, proteins, and fats, right? Fats can also be called lipids, right? Because a lipid is a bigger term, while the fat is a smaller term. That is also called triacylglycerol. We'll discuss that. So the first stage, that food that we eat is digested. The first stage is digestion, and in the digestion, which is also called hydrolysis, the food is hydrolyzed. The food is hydrolyzed to make the monomers. The carbohydrates are broken down into glucose. The major carbohydrate that is present in our diet is starch, right? That is there in the bread, that is there in the major food, that we eat, and the pizzas, burgers, all those things contain starch. And the starch makes glucose upon hydrolysis. And similarly, proteins are digested to make amino acids, and fat is digested to make fatty acids, right? And the lipids that we eat can also make cholesterol, right? So the first stage of energy metabolism is that food that we have eaten, that must have to be hydrolyzed to make monomers that are absorbable in the small intestine. So after the monomers are formed, these monomers are absorbed into the blood, and in the blood, from the blood they go to the cells. So now just an important point, only the water soluble things directly get into the blood. There's the amino acids and glucose. We can get also galactose from lactose, and we can also get fructose from sucrose that we eat, right? But the fats cannot directly, or fatty acids and cholesterol, they cannot directly go into the blood. So first they go into the lacteals. They go into the lacteals, and lacteals are the small capillaries of the lymphatic system. And once these things are in the lacteals, fatty acids and cholesterol, in the lacteals there are proteins. There are proteins, right? They're called apoproteins. We'll discuss further. And now this lipid thing, fatty acid, cholesterol, and protein make lipoproteins, and the dietary lipoproteins are called chylomicrons. Okay, fine. But ultimately these chylomicrons enter into the blood, and from the blood they enter into the cells, right? Fine. But in the second stage, what happens to these monomers? In the second stage is called the formation of acetyl-CoA. The second stage is called the formation of acetyl-CoA. Glucose makes pyruvate, and pyruvate is converted into acetyl-CoA. And similarly, amino acids are converted into acetyl-CoA. But amino acids can also take another route, that first pyruvate and then into acetyl-CoA. And similarly, fatty acids are oxidized to acetyl-CoA. So all the food actually is making acetyl-CoA. Acetyl-CoA is very important. And now this acetyl-CoA enters into the Tca cycle. The Tca cycle is also called citric acid cycle or Krebs cycle. Tca stands for tricarboxylic acid cycle. Now what happens? Acetyl-CoA is oxidized to carbon dioxide. I'm using the word oxidized. This is really very important. Acetyl-CoA is oxidized to carbon dioxide. When acetyl-CoA is oxidized to carbon dioxide, something is reduced. And what is that something? That something is NAD. FAD. FAD and NAD are reduced to FADH and NADH. So these reduced substrates are formed in the Krebs cycle. They are also formed in glycolysis and in the link reaction. We will study that further. But the bigger concept is that acetyl-CoA is oxidized to carbon dioxide, and in that process FAD and NAD are reduced to NADH and FADH2. So the product of Tca cycle are carbon dioxide and NADH and FADH2. These reduced substrates then enter into the electron transport chain and they are oxidized. And once they are oxidized, energy is produced in the form of ATP. Energy is produced in the form of ATP, and oxygen is reduced to water. Right? You see that oxidation and reduction takes place simultaneously. That is why they are called redox reactions. So if I sum this up, this becomes like food, first of all, is digested and makes monomer. Those monomers are converted into acetyl-CoA, and then acetyl-CoA in Krebs cycle makes carbon dioxide and reduced substrate, and those reduced substrates go into the electron transport chain to make ATP, right? So this is the whole process, and from here to here we can say that the food is oxidized to carbon dioxide, and in that process of oxidation we got ATP, while the first process is essentially hydrolysis or digestion. So now you know that this is the big picture. The food, all the food that we have eaten, that has been digested first, and then after digestion that is absorbed into the blood. In case of carbohydrates and proteins, directly absorbed into the blood, and from here it goes to the liver through portal circulation. While in case of fatty acids and the cholesterol or all the grease that we take in, that is first funnels into the lymphatic system, and from the lymphatic system it makes chylomicrons, and that those chylomicrons go to the blood, and from the blood they enter into the cells like other nutrients. And then once inside the cells, they are converted into acetyl-CoA, and acetyl-CoA enters into the Krebs cycle to make carbon dioxide, NADH, FADH2, and those NADH and FADH2 are oxidized in the electron transport chain to produce ATP, and when they are oxidized, oxygen is reduced to water. So this is the big picture. So how this is going to apply in the real scenario? Now comes the big picture. You should be familiar with the last slide. We cannot move forward if we don't know what we have discussed in the last slide. So here, this is also called the well-fed state. Well-fed state, we have eaten a lot of stuff, and that lot of stuff after digestion is now in the blood. What is outside the box? Outside the box, I mean, all these things that are outside the boxes, they are in the blood. So outside the box is blood, right? Now in the blood we have glucose, we have amino acids, and we have chylomicrons. These are the things now which are there in the blood after digestion. The glucose, chylomicrons, amino acids, so all those things now they are in the blood. So let's see what happens in the well-fed state. An important thing is that the well-fed state is controlled by a hormone which is called insulin. This is very important. All this well-fed state is happening under the influence of insulin, right? So let's discuss first the liver. All that glucose that is coming from small intestine enters into the liver. What happens to this glucose? Let's see. This glucose, first of all, makes pyruvate. Then pyruvate makes acetyl-CoA, and acetyl-CoA makes carbon dioxide and ATP in Krebs cycle and electron transport chain. So the first thing, right? First of all, that glucose that is entering into the blood, into the liver, that is entering into the liver, that is going to complete the energy requirement of the hepatocytes. Once the energy requirements are full, but the glucose is still entering, that glucose, that extra glucose, is converted into glycogen. So this is the second thing that is happening to the glucose. That glucose is converting into glycogen. Now the glycogen stores are full, but we have eaten a lot of stuff. So a lot of glucose is entering. Energy requirements are met. Glycogen stores are full. Now what this glucose is going to do? This is going to make again acetyl-CoA. And now there will be excess of acetyl-CoA in the liver, but we don't need more energy. So that acetyl-CoA then is converted into fatty acid, right? So this is the third thing that is going to happen with the acetyl-CoA, and that is coming from glucose. And this is the fourth thing that is happening to glucose, and that glucose, extra glucose now that is there in the liver, is going to make cholesterol. So the four things that can happen to glucose are: they're going to complete the energy requirement, they're going to make glycogen, and if more glucose is entering, that is going to make fatty acids and cholesterol, right? So this is what is happening to this glucose when it is in the liver. Now we see what happens to this glucose in the muscles. In the muscles, the same story. That glucose is going to complete the energy requirements, right? That's going to make the energy requirement, and an extra glucose that is coming into the muscles, that is going to make glycogen, right? So this is glycogen. And now we see what happens to that glucose that is entering into the adipose tissue. Again, in the adipose tissue, that glucose is going to make carbon dioxide and ATP. But another important thing, this glucose in the adipose tissue is now making glycerol as well. This is going to make glycerol as well. So this is very important point. Note it, right? That glucose that enters into the adipose tissue, that makes glycerol as well. Now we see the two organs that are essentially dependent upon glucose, and they are brain and red blood cells. Brain and red blood cells. Brain is essentially living on glucose. Glucose enters into the brain, it makes pyruvate, it makes acetyl-CoA, it makes carbon dioxide and ATP. So all the energy requirement of the brain is met by glucose. And similarly, that glucose inside the red blood cells makes pyruvate, and in that process it makes just small amount of energy, 2 ATP molecules from one glucose, and this is very important. But here you note that the pyruvate is not being converted into acetyl-CoA, rather it is being converted into lactate. Why is it so? Because red blood cells don't have mitochondria. Red blood cells don't have mitochondria, and mitochondria is essential for electron transport chain and Krebs cycle. No mitochondria is there in the red blood cells, so pyruvate cannot be converted in the red blood cells to acetyl-CoA or to Krebs cycle, or can go to the Krebs cycle or electron transport chain. So this pyruvate then follows the anaerobic route and it makes lactate. That lactate then enters into the liver, and in the liver it is converted back to pyruvate, right? This conversion of lactate back to pyruvate in the liver is called Urea cycle. It is called Urea cycle. Corey was a scientist who discovered this phenomenon. Like all the lactate that is produced in the cells due to anaerobic respiration, that goes to liver and makes pyruvate again. Now you also see that this cholesterol makes bile salts and the bile salts make bile. Fine. So this is how the well-fed state is going, and all these processes are happening under the influence of insulin. Now we see what happens to all those chylomicrons. The chylomicrons that are there in the blood, they are broken down by an enzyme which is called lipoprotein lipase. Lipoprotein lipase, and this is really very important point to note. Lipoprotein lipase breaks the chylomicrons, and chylomicrons then makes fatty acids, and those fatty acids are dumped into the adipose tissue. And again, another thing that is formed from chylomicrons is glycerol, and then that glycerol goes to the liver. And we saw earlier that fatty acids are also there in the liver being synthesized from acetyl-CoA. Those fatty acids combine with glycerol to make fat, but fat cannot be exported out alone. It needs to be bound with proteins, so it makes lipoproteins, and those lipoproteins are called very low density lipoproteins. Very low density lipoproteins, and those very low density lipoproteins are again broken down by the enzyme LPL, lipoprotein lipase, to make fatty acids and glycerol. Glycerol recircles into the liver while fatty acids are dumped into the adipose tissue. Right? Now these fatty acids, which are in the adipose tissue and coming from either the diet or from the liver, these fatty acids combine with glycerol. They combine with glycerol to make fat. Now you see what is happening. All that glucose that we have taken in, all that starch that is there in the blood is essentially making fat. You see that this glucose is making acetyl-CoA, acetyl-CoA is making fatty acids, fatty acid is making fat, and fat into VLDL, and VLDL to fatty acids, and fatty acids combines with glycerol to make fat. That is the reason when we eat a lot of rice, we get fat. When we eat a lot of rice, rice is starch, and that starch makes glucose, and that glucose is essentially making fat. This is how the biochemistry is interesting. We understand the metabolism, that's what is happening, and all this thing is happening under the influence of insulin. Like insulin lowers glucose level by pushing the glucose from the blood into the cells. Now see what happens to amino acids. Amino acids enter into the muscles. Inside the muscles, amino acids make proteins, right? And now these amino acids also enter into the liver. And in the liver, they are deaminated. Their amino group is removed, and that amino group makes urea. We'll study that urea. And the rest of the amino acid now makes pyruvate, right? So this is the big picture of what is happening to the glucose or amino acids or chylomicrons that are entering into the blood after digestion. And this is really very important. In this one diagram, we have studied about 11 to 12 pathways. And what are those pathways that did in that well-fed state? Glycolysis, like conversion of glucose to pyruvate, oxidation of glucose to pyruvate, and then pyruvate makes acetyl-CoA, and that pyruvate to acetyl CoA is called link reaction. And then that acetyl CoA enters into the Krebs cycle. So this is the third step that we studied in the big picture. And in the fourth step, the products of citric acid cycle enters into the electron transport chain. Then we saw that glucose made glycogen. So glycogenesis, we studied glycogenesis. We can see that glycogenesis happens in the well-fed state. It cannot happen in the starving state. It can happen in the well-fed state. And which hormone is going to influence this process? Definitely insulin. And then we also studied that the extra acetyl CoA that is accumulated in the liver makes fatty acids. So we will study the fatty acid synthesis. And we also know that extra acetyl CoA that is in the liver is going to make cholesterol. And from the cholesterol we made bile salts. So bile salts, will study the bile salt synthesis. Inside the muscles, we see that amino acids make proteins. So we have studied protein synthesis. And then we saw that the amino acids that are entering into the urea cycle, they're entering into the liver, they are making urea. So we'll study the urea cycle. We will study the urea cycle, right? This glycolysis is not here, right? We have already discussed. So then we'll see that the fatty acids that are either in the form of chylomicrons or in the form of VLDL, they make fat in the adipose tissue. So we studied the fat synthesis. Again, fat synthesis is happening under the influence of insulin. And then we also studied that lactate that is produced by the anaerobic respiration in the red blood cells or in other cells that is converted back to pyruvate that is called Urea Cycle. And then we also studied the fat synthesis in the liver, and then that fat makes very low density. We need to know what is happening. Glycogenesis takes place in liver and in the muscles. Fat synthesis takes place both in the liver and in the adipose tissue. But all that fatty acids that is produced in the liver, that doesn't remain in the liver, but it is exported out of the liver. It is exported out of the liver in the form of VLDL, right? And another point to remember is that glucose is necessary for the survival of red blood cells and for the survival of brain. This was the well-fed state, and there was one hormone that is controlling all this well-fed state, and that is called insulin. That is called insulin. If someone asks, like, which hormone is important for making glycogen? Insulin. Which hormone is important for making fatty acid? Insulin. Which hormone is important for making cholesterol? Insulin. So insulin is a synthesizing hormone. Insulin is important to synthesize the things, right? Now we see what happens when we are in the fasting state. So we are in the fasting state, but notice here, in the fasting state as well, the liver, the brain, and the red blood cells, there is same thing that is happening in the liver and in the red blood cells, whether this is the well-fed state or the starving state. That glucose entering into the brain to make carbon dioxide and ATP, that glucose is making ATP and pyruvate, and then pyruvate is making lactate, and lactate is making pyruvate again through Urea Cycle, right? Urea Cycle. This is the same in the well-fed state and the fasting state. But now you see the arrow of the glucose is not inside the liver. Rather, glucose is now exported out of the liver. And there is not one hormone that is doing these things. The most important one is glucagon. Glucagon is important for fasting. Cortisol is important for fasting, right? Epinephrine and norepinephrine is important for fasting. Then we have thyroxine. That is important for, T3 and T4, thyroxine and triiodothyronine, T3 and T4, this is T4 and T3. And then another hormone, growth hormone, also is important for the fasting. So you see that there are a lot of hormones that are there in the fasting state. Why is it so? To give glucose to the brain and red blood cells. because brain and red blood cells, whether this is fasting or whether this is well-fed state, follow or gets only glucose for their energy purposes. And if we are not getting glucose, then this is going to create a lot of trouble. So remember, hyperglycemia, no problem, person will survive, but hypoglycemia is going to shut down the brain and red blood cell. So hypoglycemia is more dangerous than hyperglycemia. So now we see how this glucose is exported out of the liver. How this glucose is exported out of the liver, right? So formation of glucose from non-carbohydrate sources is called gluconeogenesis. Formation of glucose from non-carbohydrate sources is called gluconeogenesis. We'll study that, but before we study that, we just see that what happens. First of all, that glycogen that was there stored in the liver, that is going to make glucose, and then that glucose is exported out of the liver. But glycogen stores are not going to last for long. They are going to be depleted. So once the glycogen stores are depleted, we need again glucose. But from where we will get glucose? We'll get glucose from alanine. Alanine is amino acid. It's glucogenic amino acids. And from where we are getting alanine? Alanine is coming from all the amino acids, right? So there are actually 18 amino acids that make alanine, right? They are all called glucogenic amino acids, and these amino acids are coming from protein. So protein is broken down in the fasting state, and then that protein makes amino acids, and the amino acids ultimately make alanine, and then alanine enters into the liver, and again it is deaminated to make urea and pyruvate, and then that pyruvate makes glucose, right? So formation of glucose from alanine is gluconeogenesis, and the formation of glucose from non-carbohydrate sources, this is called gluconeogenesis. Similarly, we can get glucose from glycerol phosphate, right? And so the second precursor for glucose synthesis is glycerol phosphate. And from where this glycerol phosphate is coming, you see that this is coming from adipose tissue. This is coming from adipose tissue, right? So fat. This is coming from here. It's making here, and that's coming here, right? And now another thing that can make glucose is lactate. So lactate is making glucose. So there are three things that are making glucose: alanine, glycerol, and lactate. They are also called the gluconeogenic precursors, or the precursors of gluconeogenesis, right? So now we see how liver, muscles, adipose tissue complete their energy requirement in the well-fed state, in the fasting state. Sorry. So first of all, things starts from fat, adipose tissue. Inside the adipose tissue, there is fat. And the fat is broken down by hormone-sensitive lipase. Hormone-sensitive lipase. Right. This is an enzyme which is stimulated by glucagon cortisol, and this hormone-sensitive lipase breaks this fat into fatty acid and glycerol. That glycerol goes to the liver and makes glucose. Now these fatty acids are oxidized. These fatty acids are oxidized to make acetyl-CoA, and then that acetyl-CoA is going to complete the energy requirement: carbon dioxide and ATP. Carbon dioxide in the Krebs cycle and ATP in the electron transport chain. And then this fatty acids enters into the blood. From the blood, it enters to the fatty acid cannot travel alone, so it needs a binding protein, and that binding protein is albumin, right? So fatty acid goes to the liver. In the liver, it is again oxidized to make acetyl-CoA, and that acetyl-CoA is going to make carbon dioxide and ATP. And another important thing that is going to happen with this acetyl-CoA in the liver is that it is going to make ketone bodies. Ketone bodies are synthesized only in the liver. Note, ketone bodies are only synthesized in the liver, but they are not used by the liver. They are exported out in the blood, and from the blood these ketone bodies go to the muscles. And inside the muscle, they again make acetyl-CoA. And that acetyl-CoA in the Krebs cycle and electron transport chain make carbon dioxide and ATP. So this is the source of energy. Ketone bodies are the source of energy for muscles in the fasting state. Again, these fatty acids are entering into the muscles, and in the muscles, fatty acids are oxidized to make acetyl-CoA, and acetyl-CoA is going to make carbon dioxide and ATP. So energy requirements of the muscles are also met by the fatty acids instead of glucose in the fasting state. And the energy requirement of liver are met by fatty acids instead of glucose in the fasting state. And similarly, the energy requirement of adipose tissue are met by fatty acids in the starving state. Right? You see that this is the big picture. And in that big picture of fasting state, we again see a lot of pathways. And now we see that we studied glycogenolysis, that glycogen is broken down to glucose. We studied gluconeogenesis. Glucose is formed from glycerol, from lactate, from alanine. So gluconeogenesis. We also studied the lipolysis, the breakdown of fat into fatty acid and glycerol. We also studied the fatty acids are oxidized, fatty acid oxidation. Fatty acid oxidation, which is also called beta oxidation of fatty acids. We will see how. So the fatty acid oxidation gives energy to adipose tissue, liver, and muscles. And we also studied the urea cycle, that is same in the welfare state, in the starving state, urea cycle. And then we studied the ketone body synthesis. So ketone bodies are synthesized and then ketone bodies are utilized in the muscles. So this is the big picture. If you don't understand this big picture, it's really difficult to understand the metabolism. If you understand this big picture, how and what is happening in the well-fed state, in the starving state, and how the different organs are getting energy and from which thing. If you understand this, the biochemistry is really very easy thing. And if you don't understand, really, the things will become really clumsy and they will become really very difficult. And then they say, right, that biochemistry is difficult. But biochemistry is really very easy if you understand the big picture. What is that big picture? The first page. Like all that food is oxidized. All that food is oxidized to acetyl-CoA. And that acetyl-CoA enters into the Krebs cycle, and from the Krebs cycle, reduced substrate go to the electron transport chain to produce energy. Well-fed state, insulin is the only hormone, and that dumps all the glucose and all the amino acids and chylomicrons into the cells, and inside the cells they make different things. While in the fasting state, there are different hormones that are important to increase the glucose level in the blood, because glucose is essential for the survival of brain and red blood cells, right? And then again, in the fasting state, ketone bodies are synthesized. And here, note here, that is really very important point, diabetes. You see that in diabetes, we don't have insulin. In diabetes, we don't have insulin. So what's happening? Actually, the cells are depriving. Cells are in the fasting state. In the diabetes, there is no glucose entering into the cells. All that glucose is remaining in the blood. That is why the glucose level is very high. And cells are essentially behaving as if they are starving. So, in the diabetes, the cells are not getting energy from glucose, rather they are getting energy from the fatty acids. They are getting energy from the fatty acids. And when in the liver, the fatty acids are oxidized, they make ketone bodies. And then those ketone bodies cause ketoacidosis. They accumulate in the blood, and in the blood, ketone bodies, when accumulate, they cause acidosis. What are ketone bodies? There are three important ketone bodies, and number one is acetone. This is of least importance in terms of energy, right? But it is important in diagnosis because this gives the fruity odor to that diabetic patient or starving patient. And the second ketone body is acetoacetate. Acetoacetate, right? This is the second ketone body. And the third ketone body is 3-hydroxy or beta hydroxy 3-hydroxybutyrate. 3-hydroxybutyrate. These are the ketone bodies. And you see that 8, right? Whenever there is come 8, that is an acid, acid or 8. These are the same thing. Acetoacetate, 3-hydroxybutyrate. So these are the acids in fact. And when the acids are there in the blood, that is going to cause acidosis, right? I hope now you understand this big picture. And if you don't understand, watch this video again and again. First clear these concepts, and then this will become really very easy to understand all those pathways. And it will take kind of not more than 2 hours to study all those pathways, right? Thank you so much. I hope you enjoyed. Please give your feedback. Thank you. Bye bye.